Let's get started, guys. How are you guys? You good? Not bad. How was there? How was biochem? Huh? You're done? Organic, I mean. Not bad. Destroyed it. Okay, good. So I just heard that there's apparently a strike next week for the biology department or something. Usually I get emails from the chairman to tell me what to do. So we'll see what happens with that. Obviously, if there's a strike, probably we won't have a quiz. And then for tutorials are exempt. So you're just killing the lecture, the best part. Okay, I understand that you guys don't like me. It's okay. Okay, but we'll see. We'll see what yeah. No, I'm not saying that. She just told me, so I have to verify first, okay? Anyways, I'll send you an email, but apparently there was a vote for a strike next week for the tuition hike for the English universities. Yeah. Anybody except you that's maybe international student here? No? Oh, right there. How do you feel, buddy? Are you ready? I'm actually, this is sad because I just became professor. So after many years, so this guy is kind of threatening my career here in Quebec, but that's okay because there's millions of universities in the world. I'm okay with moving from here. But I don't think he's going to get far with this because it's kind of discriminatory and we can get into the argument, but we're not going to do that. It's sad. It's really sad. It's affecting a lot of professors, and me, I can move and teach at UDM, right? Because I speak perfectly French, better than English actually, and so I even speak better French than this freaking minister. But anyways, what did I want to say? So yeah, so me, I'm starting to think if actually it goes through, the rate of students will decrease, and then obviously I'm the newest here, so see you later. Right, so I have to think about that. But yeah, this is sad. I don't think it's gonna go through because he's being sued on many levels for like immigration by the immigration minister. He's being sued in the education system by McGill and Concordia, and there's a lot of what do you call it? A lot of manifestation, whatever. How do you say it? Protests for the kids in elementary school. And the best part is that he was a Minister of Education. So that's really sad. Anyways, what did I want to say? So we'll see next week for the tutorial quizzes and all that. Still didn't get the grades for the exam. Should have just done it by myself, but it's okay. Did you get 266? Not yet? Okay, you don't want to get it. Good. What else? What did I want to say? So let me show you this thing here. This is the course outline. Okay. As you can see, we're a bit late on the material, and that's because we had the. So what happened? I had to move the exam, which disrupted the flow of the chapters, and so I took the time to reset the clock. Okay. And so I guess we're not gonna get to the last chapter. Probably not. And then if there's a strike, I don't know what the hell is going to happen. So that being said, we have, if that happens, we have like 4 chapters left. Wait, how many? So next week, this, 2, 3. What? Forgot that. Okay, there's 2 chapters left. Yeah. But at least, I mean, genomics is interesting. I would have liked to do it. Maybe I'm going to skip this and then go to genomics. Anybody cares about population genetics? Okay. Sorry? Okay. I can do that. We'll see how we do this. Anyways. Sorry? You vote to, that's one that's necessary. Yeah, this one we have. These 2 are like the extra 2, you know, to This is mandatory, kind of. Okay, so that's that. Any questions from me? Yeah. So they have a makeup date. I think it's on the makeup date, which is April 16th. I don't think we're going to use it for the lecture. Probably, I don't know, man. There's too many things moving this time. For sure, we're going to have less material. That's for sure. And now what do we do with the makeup date? I don't know. We'll see. We'll see by then. Okay? I'll have more information next week. Less work for me. No, I'm kidding. Okay. Chapter 7. The last part of chapter 7. There's a few parts that I skipped in the book. So that means you don't have to cover those. Okay? So just whatever is in here for now. Okay? So it's going to be somewhat quick. Same as last time, hopefully. I'm going to just start by going back over some terminology to not get you mixed up, because I think I went too fast on that. You probably know this, but just to clarify it. So nucleic acids, when we talk about nucleic acids, we're talking about the polymer, so the chain of smaller molecules, which are called nucleotides, okay? And nucleic acids, there's two types, RNA and DNA. We'll see RNA next week. This week we're covering DNA, okay? The nucleotides, there are 4 different ones. This whole thing here is a nucleotide, okay? And it's called a purine, specifically the deoxyadenosine 5 monophosphate, okay? And there's 2 other variations of this, the DATP and DADP. And then you have the same thing for guanine. So just remember, purine has less letters, more cycles. And then pyrimidines, more letters, less cycles, okay? It's important to remember this. What else? So that's for that. Now there's another term that might mix you up, is the difference between nucleosides and nucleotides. So nucleotides have a phosphate group that is attached to the sugar, while nucleosides don't have the phosphate. You'll probably see these in the book, or I don't know where, but just remember this, okay? What else did I want to say about this? I think that's pretty much it. Okay, one last thing. I know you know this, but just to reiterate. So cytosine binds to guanine through 3 hydrogen bonds, making it a stronger binding than these 2, the adenine and thymine, which have only 2 hydrogen bonds between them. Okay, this is important. We'll see why in DNA replication in a second. You'll see that in the so this week, the week after, and the 3rd week, you'll have a lot of proteins to remember, a lot of names, okay? Unfortunately, there's no other way to do it, but it's important that you know them by heart, because you'll be asked on the exam different questions referring to those proteins. I mean, if you are done with studying, you can just go while you're working and look for what the hell is a DNA gyrase, or gyrase, whatever the way you, but now you have to learn them by heart, okay? Pretty useless, but c'est la vie. So the first thing we have to talk about when we're talking about DNA replication and polymerization is the structure of the DNA, how it opens up, okay? And scientists just use this autoradiograph to see the structure of the DNA, and it's just based on radioisotope labeling, okay? So when bacteria's DNA is circular like this, and it has 2 strands, actually it's a little bit more messed up, but let's just assume it's like this. So when you need DNA replication to happen, there's a fork that opens up at specific sites. In terms of bacteria, it's called the origin site, or the origin of replication. And so when it opens up, you'll see that it will form this kind of structure here, and the fork is right here. And then DNA polymerase will come along this strand here, and make sense? So how does this DNA structure open up? So you can imagine it here on the side as a string or 2 strings that are coiled on top of each other. And if you can just imagine it in your head, if you move the 2 strings like this, it will kind of rotate on itself, right? So one thing that happens when the DNA opens up, these supercoils form, and you can try it at home with any cables. Just put them around each other and pull like this. You'll see that it will do these coils, which then become supercoils. Okay, these structures here is a problem because it prevents the DNA from being replicated, right? So there's a way to deal with this that the cells have. But before we get into that, let's just see how the DNA double helix opens up. So there's this protein called helicase, which opens up the two strands from each other. The helicase is a hexamer, so it's a protein with 6 copies of the same peptide or the same subunit. Okay, that's why it's called a hexamer. We'll see it later on again, but you have 2 types: homomer, which is the same protein being replicated 6 times, and heteromer, which is different proteins. Okay, in this case, it's 6 proteins that are the same, making the helicase. Okay, so when the helicase rotates around the strand, it kind of opens up here at the fork and just keeps pushing along the replication fork. What happens behind it is the binding of these single strand binding proteins. So these are just small proteins that prevent the 2 strands from coming back together or from getting messed up and forming different structures. Because when you open up the DNA like this, what will happen is that it can also just find a similar sequence right here and just stick to it, because DNA is very sticky. Okay, so you need proteins to prevent it from rebuilding the strand or from forming a different structure that you don't want. Now, to take care of this supercoil here, there's another protein called DNA gyrase or gyrase. I'm not sure how you pronounce it, even after 12 years, but it's okay. And it basically will cut the strand into 2. And when it cuts it here, what happens is that the tension on the coil just is released, and then it goes back to normal. And then it also has a second function, which is to stick back the DNA molecule together. Does that make sense? Easy? Okay. So if we zoom in on this, the helicase will break the hydrogen bond. So you can imagine the 2 strands here, the anti-parallel strands here. You have the 5' to 3' and 3' to 5' bound to each other with these nucleotides in between. So the helicase, all it does is that it breaks these hydrogen bonds in between here to open up the structure, while the gyrase will actually cut the phosphodiester linkage, which is between this carbon and this carbon. So basically 3 and 5, okay? To remove this phosphate group and then bring it back together. Make sense? Okay. So the DNA gyrase is a type of enzyme that are called topoisomerases. DNA gyrase is one of them. It has a nuclease activity, important term. Nuclease is the same thing. It's analogous to peptidase or lipase or whatever. It breaks nucleic acids. So nuclease breaks nucleic acids. And that's it for that. So the main enzyme for DNA polymerization is called DNA polymerase, obviously. And it was discovered by this Kornberg guy, and the first one that was discovered was polymerase 1. Now we'll see that polymerase 1 is not the most important one for this process. It has a role, but it's not the main one. The main one will be DNA polymerase 3. This is the enzyme that is going to take care of replicating the DNA. Okay. There's also 2 others, polymerase 1 and 2, DNA polymerase 1 and 2, which have different functions. We'll see them in a second. And then obviously next week we'll see RNA polymerase, etc. So when we put DNA before the word polymerase, it just means that it requires DNA as a molecule to do the polymerase reaction. If we were talking about RNA polymerase, it acts on RNA polymerase. I know you guys know this, but it's important to reiterate. On this picture here, you'll probably see a lot of these pictures. It's just a protein structure with the beta sheets and all that. You'll see that in biochemistry. So we'll start with bacteria, and then we'll go to eukaryotes, because DNA replication is somewhat similar. It's just the name of the proteins that change and the number of them usually, okay? So in bacteria, what happens is that there's a protein called DNA-A, which will recognize the origin of replication. So if you remember, when we're talking about the plasmid, there's usually an origin of replication. Sometimes there's two, but usually just one. And this DNA will find this origin of replication and bind to it. It will also bind to the AT-rich. So this is a section on the DNA strand that has a lot of ATs, so adenine and thymine. And it will bind also at other places like the DNA boxes, okay? And this is just to create a structure that will be important to create the replication fork. Now, when these subunits of DNA, when these units of DNA A bind to the DNA, then they will allow for the recruitment of DNA B. And DNA B is just a helicase. So it will be recruited right behind the DNA A, and then start rotating around the strand and pushing away the DNA A proteins. Does that make sense? Okay. One important thing is that there are two helicases, one on each strand, and they go basically in opposite directions until they complete the whole chromosome. Okay. Now, weird enough, these DNA B are recruited at AT-rich regions. And the role of DNA B, or the helicase, is to break the hydrogen bonds. So for some reason, the cells were able to evolve in a way that the helicase does not go to GC-rich regions, but to AT-rich regions. Okay, so it's easier to break. When these helicases start rotating around the strand, they will remove the DNA A proteins, and then the DNA polymerase will be able to be recruited because there will be space for it, and it will come right behind the helicase. It will be bound to something called a beta clamp, so this gray little thing here. And the beta clamp, all it does is that it keeps the DNA polymerase 3 bound to the strand, because DNA polymerase 3 is actually not, does not stay a long time on the DNA by itself. It's a disruptive, forgot the name of the enzyme, but basically it does like 10 different nucleotides and then gets off, okay? So the clamp keeps it there so that it can undergo like a longer thread of like 1,000 base pairs, 1,000 nucleotides or something like this, okay? Just remember properly that DNA polymerase 3 is the one that does this, okay? Not 2, not 1. What else? I think that's pretty much it for this. Am I going too fast? No? Yeah? Yeah, exactly. Just one thing, the DNA A does not go just to the AT-rich region. It goes to the DNA A boxes also, okay? The role, mostly it is to stabilize the structure. That's it, okay? Okay, so DNA replication is very accurate, but for some reason you can see that we're all different. So it's still sufficient to, so the mutations that happen are still sufficient to create this much diversity, and obviously it took many years of evolution. But the reason why the DNA replication is accurate, it's because the polymerases have another function. So the first function is to copy the DNA, but the second function is called an exonuclease function, and it happens in the opposite sense. So DNA replication happens from the 5' to the 3', and I'll get into this in a bit. The exonuclease activity goes in the opposite sense. So it serves as a proofreading mechanism. The only one that also has a 5' to 3' exonuclease activity or proofreading activity is polymerase 1. And we'll see what their roles are, all three. So what happens is that when the DNA polymerase is copying the DNA, it will kind of go backwards, detect if there's a problem, and can fix it and continue. So it kind of just goes like this. right? Until it finishes the whole thing. If it finds a nucleotide mismatch, it will cut it and then replace it. And this looks like this. So again, just to reiterate, the polymerization process, which means the addition of nucleotides to the strand, happened from a 5' to a 3'. So if you have a template strand like this, 3' to 5', it will start from here and then go this way. Okay? It's important to remember that the 5' to 3' is referring to the new strand, not the template strand. Okay? That makes sense? And this is done by DNA polymerase 3 because it's the one that can go for 1,000 base pairs nonstop. Now, the repair mostly happens by DNA polymerase 1 and 2. They will come right behind DNA polymerase 3 and start checking everything. And what they do is that they start translocating. They see that there is a mismatch here. They cut it and then continue going on. 3' to 5' exonuclease activity means that the DNA polymerase 1 and 2 will go in this direction, the opposite of the other polymerase. And so they will attach and correct backwards. Obviously, it makes sense because if you're polymerizing in this direction, you want to go backwards to check if everything is good, right? You could also just follow it, but just remember that it goes backwards. Okay. What else? What else? Now, these enzymes here are not bound to beta clamp, so they will do like few subunits, a few nucleotides and then get off, and then others will be recruited, etc. Okay? Good. Okay, back to the fork. So, let's say a helicase opened the DNA, and then DNA synthesis started. So when DNA synthesis starts, there's two different strands that appear. Okay, so you have the fork forming like this. You have two different strands. The top one goes from a 5' to a 3' right here. The other one goes from a 3' to a 5' right here. So when DNA synthesis is going from the 5' here to the 3' here, that's in the same direction as the fork opening. This means that the direction of synthesis going towards the fork, the opening of the fork, so you're basically copying it's what we call the leading strand, okay? And then the opposite one, when the DNA synthesis goes from the fork away from it, this is called the synthesis on the lagging strand. And there's a difference between these two. It's important to know it, because this one here, where you're copying the DNA on the leading strand, you're following the fork, right? And so when you're following the fork, it will be continuous kind of. On the lagging strand, it's the opposite. You need to wait for the fork to open up some DNA and then bind here and then go the opposite side. Wait for it to open more, bind next to the fork and go this way. This is called the lagging strand, okay? Make sense? Good. What's important to remember about this? So when you're talking about the leading strand, as I said, it will be continuous, as you can see here, right? On the top part here. This is the leading strand. Obviously, there's a primer that is formed here. It's not shown on this diagram, but you always need a primer before starting the replication, be it DNA or RNA or whatever, okay? And this primer is added by an enzyme called primase. It usually adds about like 10 nucleotides, if I remember correctly, or 12, something like that. Okay, doesn't really matter how many. It's just important to know that it will add a short sequence of DNA, and then that will be recognized by the DNA polymerase, which will allow it to attach to the DNA and start the synthesis. Just like this, RNA primer, DNA polymerase 3 comes here, binds here, and starts copying. Okay? So in the leading strand, usually there's less primers being built because DNA polymerase does not get off as often. You have a primer followed by like 1,000, 2,000 base pairs, and then another primer, etc. For the lagging strand, now it's a bit different. You have primers being built every few nucleotides, and then the strand being synthesized in this direction here. So what happens is that the fork is opening this way to the left, and then you have a primer being attached here and DNA synthesis going here. When the fork opens up a little bit more, there will be another primer added, and then synthesis going into this way. Okay. The new sequence on the lagging strand with the primer is called an Okazaki fragment, and it's just related to the guy that figured this out. Now, one more thing. The polymerization reaction happens with DNA polymerase 3. Okay, so it's 3 that's making this yellow strand here. But these primers are removed by DNA polymerase 1 and replaced by DNA polymerase 1. So this is why they need to be about 10 nucleotides approximately. It's because these DNA polymerase 1 and 2 cannot go longer than 10 nucleotides before getting off. Something like that, 10 to 12. Okay. And so you end up having multiple primers being broken down and then replaced by actual sequence with DNA polymerase 1. Pop quiz. Why is DNA polymerase getting off the DNA compared to polymerase 3? Yeah. Sorry? The clamp? Yeah, exactly. Cool. So the other thing is, when you have the continuous generation or synthesis of DNA, you don't need to stick parts together. While in the lagging strand, what happens is that when you destroy these primers, you end up with a hole here, which gets replaced, but the two strands here are not joined together. So there's another reaction that happens called the ligation. I think this is pretty obvious, but basically it's sticking the two units together. Cool. Okay. Yep. So Okazaki fragment is, first when we talk about Okazaki fragment, we're talking about the lagging strand. And so the Okazaki fragment is just the fragment of DNA and primer together, right? And then the ligation will join two Okazaki fragments together. Cool? Good. Did I say this? I think I said this. Oh, this is why it's called the semi-discontinuous synthesis, okay? Because one of them is continuous and the other one is not. What else? I think this is good. Okay, did I talk about the bond? Okay, so the ligase, all it does is that it creates a phosphodiester bond between two nucleotides. Okay. Now a little bit of chemistry, not too much, but just a little bit. So DNA polymerase, well, all the polymerases have this function, but basically their goal is just to create another, or to stick two different nucleotides together, okay? Using a template from the other side. But the reaction that happens is that at the 3' carbon of one nucleotide, it will be joined to the 5' of another one, okay? And the unit here that's being used as a precursor for the next nucleotide being synthesized is not, let's say, for example, in this case, it's cytosine, so it's not a DCMP, but a DCTP, so a triphosphate. And then the reaction will happen here and kick out the two phosphate groups, leaving a CMP. Make sense? Kind of. You don't need to know where the attack is happening and where the electrons are, okay? This is not relevant for this course. So again, you have a DCTP that is used to generate a DCMP and bind it to the nucleotide before it. And this happens in a 5' to 3' fashion. Okay. I think that's it. And obviously there's two phosphates that are lost, generating this structure here. Okay, so all this is basically this thing here, okay? So we'll go over it quickly just to see step by step what happens. So the first thing that happens is that the helicase comes in, opens up the two strands. This will create supercoils which are fixed by the DNA gyrase. This DNA gyrase, all it does is that it cuts the DNA here at the phosphodiester bond, allowing for the tension to be released from the supercoil, and then when that's done, it sticks them back together. Followed by that, there's also these SSB proteins that are bound to keep the DNA open and not, imagine your charger there, it gets all mixed up, okay? So the SSB just keep it straight, okay? And then when that happens, there's two DNA polymerases that bind, one on the leading strand, starting from here, and following the replication fork direction, which is this way, and the other one going in the opposite sense. So it would bind here, go a little bit, get off, and then this would get opened up more, bind here, and go in this direction. So if I ask you which direction is the replication fork going, which one? It's going this way, right? So 3 to 5, right? Good. And replication is 5 to 3. But not on the template. It's on the synthesis happens from a 5 to 3, but the direction of the template is 3 to 5. Okay, it's important to remember this. This is actually one of the only things that probably you'll need to remember if you're going into research or all that, because 5' to 3' is important. If you're making your plasmids, qPCR, anything. Okay, so one of the only things that like it's important to remember by heart. Okay, what else? DNA polymerase, as I said, just breaks these primers and regenerates the sequence, and then DNA ligase will stick together two fragments. I think that's pretty much it for this. Oh, and primase will make these primers. Other than that, you also have to remember that at the origin of replication, it's happening on both directions, right? So you can see it will look kind of like this. You'll have two leading strands depending on where you are, if you're going this direction or this direction, so you'll have two different forks. One going this way, one going the other way, okay? It's important to remember this. In human cells and all that, there's hundreds of origins of replication, okay? So it's going all over the place. And it looks kind of like this. This is in eukaryotes, okay? Not in bacteria. So you'll have multiple origins of replication that will open up at the same time, and then DNA replication will happen at all these origins at the same time until they reach this point where they are both joined, and then everything gets off the DNA structure. And this is what leads to sister chromatids being formed, homologous chromosomes, and all that stuff. Okay? Not bad. Okay. So, yep. Yeah, most of the time only one. I mean, there's sometimes two, but most of the time it's one origin of replication, and it's just going in both sides and until the end, right? But in mammalian cells, in yeast, for example, which is a eukaryote, you have multiple origins of replication. I think yeast has 400, if I read the book correctly, or something like that. Humans, I forgot how many, but a lot. There's like 3 billion base pairs, so you can imagine that. Compared to the few millions of bacteria, it's a lot. Okay, cell cycle. This was the topic of all my research from undergrad to now, okay? When you're studying cancer and all that, you'll see this a lot. So the cell cycle has 4 main phases. The G1, G2, M phase. Who already seen this? Some people? Okay. There's another state, the G0, which we're not going to talk about, but that's basically the state in which your cells get in. They don't divide anymore, and that's called senescence, and that's when your cells get too old, okay? This is one of the important parts of aging, and it's being studied all the time. If you're studying cancer, most of the time you'll be focusing on the M phase, which is the mitosis or meiosis phase, because this is where cell division happens. We are interested in the S phase. So G1 and G2 are they're called gap phases. Nothing really crazy happens here. The cells are just in a relaxed state until they get the cue to start dividing. When that happens, DNA will be replicated at the S phase and then segregated into two cells during the M phase, meiosis, mitosis, whichever. Okay, so in the S phase, this is where all the polymerization happens, right? It's also called the synthesis phase, obviously, S phase. That's just because there's a new DNA molecule being formed, okay? So in this case here, one important question that we have to remember is how does the cell know that it needs to divide, right? Because some cells in your body right now are dividing, other are not, and it's just because it's getting cues from different cells, right? If, for example, you work out at the gym and you're eating a lot of proteins, your body will get this signal from the proteins telling it it's time to start growing muscles, and so the cells will start dividing, right? If you are starving, the cells will receive the signal of we can't divide because we don't have enough nutrients, okay? And some of these factors all converge on a few different proteins called the cell cycle proteins, which you'll probably see in the future a lot, especially in cell bio and advanced cell bio. But in our case, this is just general information. We only care about two of them, CDT1 and CDC6, and I'll tell you why. CDT1 is CDC-dependent something protein, okay? And then there's CDC6 too, okay? So these proteins here will tell the cells to start copying DNA. If you don't have these or there's a mutation in these, then their replication does not happen. So you can now imagine a pathway where you have too much protein intake and your body just receiving the signals that will activate these CDC6 and telling it it's time to grow, okay? And there's other different factors that affect these proteins. But in our case, I think that's it. Yeah, they act as activators. That's pretty much it. In our case, we're only interested into these two. So for eukaryotic cells, the replication happens quite similarly. There's a protein complex called the ORC, or origin of replication complex, that will come and bind to the DNA at some consensus sequence. So this is just some sort of sequence on the DNA that this protein complex recognizes. When you hear protein complex, just know that it's multiple proteins together. But it's basically the same thing. This ORC binds to the consensus sequence. The only difference is that there's this CDC6 protein that comes and activates this ORC, which changes its conformation, allowing for this helicase to come and bind to the AT-rich region. And even the helicase needs to be activated or primed by the CDT protein, CDT1 protein. So this is why these two are important, because they basically tell the ORC that it can do its job. If these were not here, then these proteins would be useless. They would bind, but they have no role, okay? So these are activators. Did I forget something? Again, you have the same thing, the AT-rich region. This is where the helicase will start rotating around. So when that happens, there's a second helicase that comes to go in the opposite direction, of course, on the other strand. And then you have the recruitment of the DNA polymerase that's bound to the beta clamp. And that's pretty much it, okay? Now, one important thing to remember is that the S phase starts from these helicases, and then these recruitment of these proteins here starts in the G1 phase, okay? So the signal to start replicating happens in the G1 phase, but the replication happens in the S phase. Okay, good. What else? Right, so in the case of DNA replication what happens is that when you're dealing with the lagging strand, you'll have primers added and then replaced by DNA polymerase 1, etc., right? But there's the last primer there that when it gets degraded, it does not get rebuilt. And so it creates a 3' overhang. 3' is important here, okay? It creates a 3' overhang. And this 3' overhang technically would get degraded by nucleases in the cell because it's just hanging there. Now you can imagine that if the cell starts degrading its own DNA, there will be a lot of problems, okay? Actually, this does happen through time. So the cell is able to protect itself, but to a certain limit. But what happens is that these overhangs get degraded, and then your own DNA gets degraded, and then at some point your chromosomes will be so destroyed that your cells will start dying. Okay, this is one of the first found and major contributors to the aging process. Okay, so aging, if you're trying to figure out, there's 9 or 12 different hallmarks, but this is one of the major ones. Okay, these, yep, exactly. So this is called the telomere. This little overhang here is a telomere, and it's just meant to protect the DNA from being degraded, right? But obviously, it will get degraded at some point because the machinery to protect it will become too old due to some physics stuff, and basically kill the cell. Okay, make sense? Enzymes, unrelated to this, but for some physics people. Technically, if there was like no entropy and all that, you would be living forever, right? But because everything decays, it becomes less efficient. Then what happens is that this protective mechanism that is built for you will be too weak to repair this kind of stuff, okay? And then degradation happens. I think I said it properly. Sorry? Makes sense? Yeah. I fight all the time with my dad for physics stuff. But, people fight about, movies and all that. We fight about physics, Okay, so these overhangs are called telomeres. And the cells have this enzyme that can repair them and lengthen them, make them longer. So this enzyme is called a telomerase, and it will come and bind to the overhang and then extend it, make it longer so that when degradation happens, it does not touch the actual genes that make the proteins, okay? The telomerase has a specific function that is a bit different from DNA polymerase. It's called a reverse transcriptase. So DNA polymerase takes DNA and makes DNA. RNA polymerase takes DNA and makes RNA. Reverse transcriptase does the opposite of RNA polymerase. It takes RNA and makes DNA. Okay? And so this telomerase here will use a template RNA molecule. So you have this green thing here is the telomerase with this strand here. Okay? And when you have an RNA molecule bound to a protein, we call it a ribonucleoprotein. So RNP. Okay, this is a type of proteins that exist in the body, and it will use this template to create a longer telomere. Okay, and it does this in few steps. First, it will bind to the overhang, and then it will use this template RNA molecule to remake the DNA, and it will just start copying it. Okay, and sticking it together, and then it will translocate. So then there's another hole being created. You see here the blue thing here, the 3', there's nothing. And then it will recopy it and then keep moving along the DNA. Does that make sense? Good. There's a lot of research happening on the telomeres. There's a few species that, like there's a mouse that can live for like 28 years, which is a lot. Usually they live for like 3 years. So it's being studied, and it almost doesn't get cancer, right? So it's being studied to see how it could affect us and could help us prevent cancer and all that. And one of the important things that they have is that their telomerases are super strong. Okay, there's different levels of proteins, but their telomerase can make longer stretches of DNA, okay, to protect the DNA itself. What else? So after this telomerase moves along the strand and makes this whole DNA strand, what happens is that there's a hole that's created here on the opposite strand. And so this is just basic stuff. There's a primase that comes, makes the primer here, and then DNA polymerase recognizes this primer and then just starts rebuilding the strand at the top. Is this clear or? Okay. What else? What else did I want to say here? I think that's pretty much it. And obviously, there's a ligase that binds these two fragments together because you have an overhang here and you're making a new sequence, so you need something to kind of bind them together here, and that's always going to be a ligase. Now, in animals, the telomere sequence is just a repeated sequence of TTAGGG. Depending on the species, this changes. Not of humans, but of animals, this changes. And so it's just a useless sequence, okay? You've probably seen reverse transcriptase in the past, or at least heard about it. Does anyone know what it is before today? Okay, COVID. So let me tell you about COVID. If you remember properly, there were two different tests. Either you had a blood test or that little swab there that destroyed your nose. Okay, the swab there, yeah, I had some bad experiences. The swab there will just get basically cells from your nose, and then you can extract the RNA from them, okay? When you extract the RNA from there, obviously you will get both the viral RNA and your own RNA, and you can use reverse transcriptase to make a DNA molecule from that. And why would you do that? Because RNA is very, its half-life is super short and it gets degraded pretty much anywhere. So you make a DNA molecule from that, and then you can use it to see if a specific gene is there. So COVID, it was an RNA virus. It had RNA in its shell. And so when it was sequenced, the way they did it, they took the RNA, converted it into DNA, and then went and sequenced that. So now you know the sequence that is there. Then what they did is that they took that sequence and you create a primer from it that is specific only to the RNA, to the DNA of the virus that you would not find in our genes. So then when you run it into a machine, it will tell you if you have viral particles, if you have the virus in you, because it's able, you would be able to recopy that DNA strand, right? And the more you recopy it, the more light it makes in this machine, and you know that it's found there. So this is why this test was much better than the blood test, because it does not need to wait for the viruses to start being made, the proteins and all that. You can just take directly the genetic material and detect it much earlier, and it's much more precise. This is also the technique that's used to detect HIV later in life, because HIV is also a retrovirus, and on top of that, it has its own reverse transcriptase, so it's able to turn into RNA into DNA, and it does this into your body, and that DNA integrates your own DNA. Okay, this is why it's super dangerous as virus, and we have to be careful with it. Okay, cool. Another disease, Werner syndrome. If you realize, a lot of the DNA problems will lead to aging, because it's like the ultimate cell defect. And this Werner syndrome here leads to premature aging. There's a few others, and it's caused by defects in the DNA repair machinery. So in your body, you have molecules that repair DNA. If anything over here messes up, where's this thing here? Here messes up, it will probably lead to a defect, because if you don't have the DNA, it's a disaster. And this Werner syndrome, all it does is that there's a few years of difference here. It's not like this one is like 10 and this one is 90, okay? This one is probably like 40 years old, okay? And so what happens is that because you have a defect in DNA replication, you end up showing up a lot of the phenotype from old people. Weird enough, in the US, this happens once in 200,000 people. In Japan, it's 1 to 20,000 people. But what's even more weird is that in Japan, they have the populations that are the longest living. So they have like the two opposites, right? So they are being studied. The people that participate in the studies, okay, there's a lot of aging and anti-aging research happening in Japan, okay? Because there's something that they have that allows them to live much longer than us. Yeah. Yeah. Sorry? No. Okay, wait. Let's. Okay. So they don't all work. There's some of them that work, and usually those that work, you need a prescription for. Okay, let's start by that. Why they don't work? Because the products that you see on the pharmacy, if you go and take the box there, look behind it, you'll see a number, the NEP number, natural extract product or natural health, NHP number, okay? If you see that there's an NHP number, it means that it was created by pretty much any company. It could be even Pfizer, because they make stuff like this, Johnson & Johnson, but they did not go clinical trials, okay? So the process to approve these NHP products, you know, like the vitamins and all that, you guys could start selling it, right? I'll show you actually while we're into this, okay? So Canada, NHP. So now if you go to pharmacy, look at this, okay? You can look for licensed natural health products database. If you ever heard of the fancy term called biocosmetics, which they use kind of some sort of natural stuff to make your skin look better and whatever, they are not based on research. They are just formulations created by people like you and me. Obviously, the big pharma are also involved into this, because us, the consumers, if you're not educated into this, you'll think that it's super anti-aging, it works, you know? But reality is that these things are just built from research on PubMed for all the articles, and then you see, oh, a little bit of olive oil is good for this, and then add to it this, and then, right. And so they make these mixtures, which on paper could work, but in reality, most of them don't, okay? And the only requirement for this to be approved is that when you do the application, you make sure that you cover all the possibilities for health problems and you follow the daily value maximum, whatever, okay? And you can find all the products that you have in the pharmacy right here. So you can just click here and look for, where is it? Totally unrelated, but we're almost done, so it doesn't matter. So you can look for, I don't know, brand name. Let's find a name. You know what? Let's do it. Jean Coutu. It's okay. It's important to learn this stuff. Beauty. Where are you, beauty? Maybe not skin. There you go. Skincare. Oh God, I should have prepared this before. Okay, how about this? I'll show you one next class, okay? Because I have to do some research and find the product. But most of them are not based on clinical research. That's my point. And usually the ones that are based on clinical research, you'll find the bottle at like 60, 70 bucks, okay? So that small little section in Pharmaprix there with like the small little bottles like this, okay? Those usually work because they're based on clinical trial, but not the other stuff, okay? So be careful with that. And you can, I'm not saying that those that were licensed by the NHP don't work, but most of the time it's just people like me and you making them. And you can start building those formulations and selling them to any company like Pharmaprix, Jean Coutu, Proxim, etc. Okay. Sorry? So this you're talking about the anti-aging products or the skincare? Both? Okay. Right. They do, so it's a bit hard to talk in terms of humans, okay? There are some longitudinal studies that were done for over 20 years, but it's hard to like give something to a human and then wait for him to die and see if it, you know? It's actually, this is how we do the research, right? In terms of cells, there's products that make them live longer by a factor of like 5 times, okay? But now the problem is that in humans, it's a bit more complicated because the human is not just a cell, it's a full system, right? And so there's a lot of things that come into play. On top of that, there's the environment. On top of that, there's stress. On top of that, there's the goal. So, So at the end of the day, it's going to be kind of hard to assess. Okay, but in mice and all that, there are products that do extend lifespan. The skincare bullshit, that's not okay. It's just a lie. It's just marketing, most of it. Okay. Yep. So actually, the technique that you just saw, that's called cloning. That's basically called cloning. So when you hear someone say, I'm cloning this gene, you're basically doing that pretty much. And this is the technique that was used to clone Dolly the sheep, if you ever heard about Dolly. And there's a few other clones that were built. You can Google this. Even in molecular biology, you'll learn about cloning and you'll be doing some cloning. Not humans, but some sort of strands. But it's the same thing. It's just much longer, right? That's pretty much it. Technically, it's possible. Now, if we get into religion and all that stuff, it's a bit more complicated. But technically, it is possible. Okay? Now back to this little thing here. I'll have something for you next week, so you can see. Sorry for the people that want to leave and are getting bored by my tangents, okay? It's important to have fun, okay? Now, one more thing. You can see this here. It's a list of proteins that are found in bacteria and eukaryotes. This is just the equivalent between both organisms. Learn those that I talked about. Obviously, I didn't talk about the clamp loader, so who cares? But the others know which one is which, okay? One last thing that's important for exam purposes. So let's say you see a DNA strand like this. I give you this on the exam or quiz or whatever, and it looks like this, okay? When I tell you to copy the top strand, which direction is it going to go? Forget it. Sorry? This one here? It will go from the opposite, right? So, wait, let me just show you there. Do I have a pen? Okay. So if I ask you to copy, where's my pen there? I can't even see it. Let me try this. So if I ask you to copy this strand here, it's going to go from here in this direction, okay? Because the strand being copied is going from 5' to 3', okay? Does that make sense? And then same thing for the other one. This one goes this way, okay? If I don't write, okay, maybe not me, but if someone does not write the direction, you just assume that it's a 5' to 3', left to right, same direction as we are writing. To replicate, let's say I give you this sequence and I ask you to replicate it, the way you do it in the lab, and it's the same thing like everywhere, it's basically you have to meld the DNA to make it into a single strand. You can heat up the DNA, and then you will find a primer. So if I want to copy this specific sequence, and it's a 5' to 3', then I'm going to have to make a primer on the right here. And the primer will be G, A, T, G, C. Does that make sense up to now? Then I'll add DNA polymerase, which will recognize that primer and continue synthesizing from right to left. And to do this, I need a few things. I need DATP, DCTP, DGTP, and DTTP, or dinucleotide triphosphates, something like this. Nucleotide, deoxynucleotide triphosphate. Okay, you need all 4 of them because these will be used to generate the other sequence. Now, the strand you're using is the template, right? And the other strand is the complementary strand. Okay, so if I ask you for this one, this is the template, this is the complementary. If I ask you for this one, this is the template, this is the complementary. We'll be specific. Yeah, pretty much. There you go. Okay, what else? And this is basically what I just said. So you have the primer here, and then you have the polymerization reaction happening in this direction. Cool? That's it.